Good morning, everyone, and welcome to today's MPG Primer session. I am very excited to introduce to you Jenna Ballard, who will be presenting today. She's an associate computational biologist in Luke O'Connor's lab at the Broad Institute, where she has been focused on developing and applying statistical methods to genetic data to better understand the genetic architecture of multiple complex diseases and traits. Today, she will be sharing with us her recent work on exactly that topic. She has pauses set in her talk, and so please type any questions you have in the Q&A or raise your hand, and we'll either recognize you to speak or we will voice your questions for you. So thank you so much, Jenna, for joining us today.
Thank you very much for the introduction. And thank you for the opportunity to present this work this morning. It's actually now published in AJHG, shown in the lower right corner. I'm looking forward to telling you about what I've been working on. So first, what is genetic correlation? Genetic correlation is the correlation between the effect sizes of a set of variants on a pair of traits. It indicates the presence of pleiotropy, which is when a single variant affects multiple traits. And genetic correlations close to 1 or -1 indicate that variants affect the two traits in a similar manner, and that these traits are more genetically related. So previous studies have found that there are many pleiotropic SNPs which affect multiple traits. And as shown here, cross-trait LD score regression showed that many pairs of traits are genetically correlated. However, correlation does not give a complete picture of shared genetic architecture, and pleiotropy can provide insights that might not be captured by the genetic correlation. In this toy example, on the left, effect sizes of the pleiotropic SNPs on each trait are proportional, as the effect sizes on trait 1 are half the effect sizes on trait 2 for all SNPs. Thus, the effect sizes are highly correlated, indicating the existence of pleiotropy. However, as shown on the right, there may be pleiotropic SNPs whose effect sizes are uncorrelated, as they do not affect each trait proportionally, with different relative magnitudes and directions of effect on the two traits. And an extreme case of this could be that the genetic correlation between traits is 0, even though there are actually many pleiotropic SNPs. We're interested in pleiotropy because it is mechanistically informative, allowing us to find clusters of SNPs with similar effects across multiple traits, pointing to shared pathways between those traits. For example, in the simulated data shown here, we can see clusters of SNPs that affect subsets of coronary-related traits, indicating potentially distinct mechanisms underlying BMI-related traits, cholesterol-related traits, and hypertension-related traits. The scatter plots in panels B through D display the effect sizes for each SNP on 2 traits at a time, and the SNPs are colored by their component membership. So from panel B, for example, we can see that BMI-related SNPs affect both CAD and BMI, with positively correlated effect sizes, whereas cholesterol-related and hypertension-related SNPs affect CAD only, which is also reflected in the heat map. We will see similar scatter plots later on in our real trait analyses. Also note that here the SNPs were simulated to belong to a single component, but our model actually shows SNPs, our model actually allows SNPs to belong to more than one component, which is more in line with the tagging that is observed in marginal effect sizes. So therefore, knowledge of pleiotropy is much more informative than simply the genetic correlation, and using pleiotropic decomposition regression, we aim to utilize the information gained from pleiotropy to identify the sources of pleiotropic clusters using pleiotropic components. We also aim to improve association power similar to methods such as MTAG, and later on I'll show some out-of-sample replication analyses for this. So there are some previously existing methods that already do these things, such as SVD or singular value decomposition and genomic SEM, which identify components of heritability in MTAG. And then there's also MTAG, which improves effect size estimates. We did some comparisons between these methods and ours, and I'll show some results later on in this talk. Additional potential applications of models of genetic architecture include causal inference, and Mendelian randomization is one possible method for that. So starting with the two methods that can identify components of heritability, SVD and genomic SEM. SVD works by performing singular value decomposition on the summary statistics matrix, and this is equivalent to eigendecomposition of the summary statistics covariance matrix. The resulting decomposition results in components of heritability. Another method that does this is genomic SEM, which estimates a factor model that matches observed genetic correlations and can identify clusters of related traits. It is notable that both of these methods operate on the genetic correlation matrix alone and do not model the effect sizes of individual SNPs. Because multiple decompositions may be possible, this is a limitation of these methods and their ability to find the most parsimonious or biologically informative decomposition. MTAG is a method that improves effect size estimates by performing cross-trait meta-analysis using optimal linear weights that are calculated from the genetic correlation matrix. Pooling information across multiple related traits improves the power to detect associations, and in our method, we also take advantage of the use of multiple genetically correlated traits to try to improve this even further. And then finally, there are things beyond estimating genetic correlation that we might want to do, such as infer causal relationships between traits. And one existing method for this is Mendelian randomization, which aims to test whether some trait, which we call the exposure, may be causal for another trait, which we call the outcome. Because our genetics are randomly assigned during meiosis, they're unlikely to be affected by reverse causation or our specific lifestyle factors. Mendelian randomization works by finding a genetic variant termed the instrumental variable, which is robustly associated with the exposure. This variant must not be associated with confounding factors that might affect both the exposure and the outcome, and the genetic variant must not be associated with the outcome other than through the exposure. If these conditions are satisfied, it may indicate that the exposure is causal for the outcome, or it at least provides evidence that is not inconsistent with a causal relationship. So given that overview of previously existing methods for partitioning heritability, improving effect size estimates, and causal inference, here I present pleiotropic decomposition regression, or PDR, which mainly aims to do the first two, but could have potential applications in causal inference as well. So before I go on to talk about PDR in more detail, I'll pause and see if there are any questions so far.
Thank you so much, Jenna. And none have been raised so far, and I will just encourage the audience, if there are any questions that arise to, again, please post them in the Q&A. So thank you very much, and please go ahead, Jenna.
Thank you. So PDR models the marginal effect size distribution by breaking heritability into pleiotropic components, each of which is defined by a single covariance matrix. The marginal effect size vector is therefore modeled as a sum of effects from K of these independent components. Each component follows a scale mixture of Gaussians that have zero mean and covariance equal to a random scaling variable, sigma k squared, multiplied by a pattern matrix representing some specific type of pleiotropic relationship, such as trait-specific SNPs or SNPs that affect different subsets of traits with particular correlation patterns. The scaling variables determine the proportion of SNPs that have an effect size of a given magnitude. For example, if sigma k squared for a SNP is 0, that SNP does not affect component k. The main types of components that we're modeling are trait-specific, shown by scatterplot A, and pleiotropic, as shown in scatterplot B. Trait-specific components consist of SNPs with non-zero effect on a single trait. The one in panel A is therefore trait 1 specific. Pleiotropic components consist of SNPs that can affect multiple traits with various correlations and relative effects on each trait. These components can also be visualized as heat maps, which represent the covariance matrices of effect sizes on each trait, as shown in panels A and B. So the trait-specific heat map has non-zero heritability for trait 1 only, and the pleiotropic component explains heritability for both traits, where the SNPs have positively correlated effect sizes. The marginal effect size of a SNP is modeled as a sum of effects contributed by each component, which is shown in panel C. So here's an overview of PDR. We take the GWAS sum stats and pre-process them using LD score regression and normalization to make the variance equal to 1. From this, we get effect sizes that we input to the model, as well as a noise covariance matrix. Then we calculate the empirical characteristic function using these effect sizes observed from the data, which is what we use as the dependent variable in the regression. We also build a model that we want to fit to the data using a combination of trait-specific and pleiotropic components. We have two types of pleiotropic components, factor-like and generic. The covariance matrices for the factor-like component are rank 1, and those for the generic components are full rank. We mainly use the generic components in our main analyses since they tend to fit the data better, but we did use the factor-like components in one of our simulation analyses. To fit the model, PDR has an initialization procedure where it initializes the model and does a few gradient descent steps several times, and then selects the model with the best initialization. Then using that model, it does multiple iterations of fitting the model parameters and using gradient descent and fitting the mixture weights and updating the empirical characteristic function. Finally, once we get the fitted PDR model, we can perform downstream analyses such as calculating posterior mean effect sizes using the estimated effect size distribution. So here's the estimation equation. It is a method of moments feasible generalized least squares estimator that uses time domain moment equations. It performs moment matching using gradient descent. The main thing being estimated here is the heritability explained by each component. Note that each component indexed by K is a mixture of Gaussians with a specific pattern covariance matrix scaled by a specific scalar multiple, sigma G. So there are G distinct mixture components specified by their scalar multiple. Since we allow SNPs to potentially belong to more than one component where their effects are the sum of effect sizes across components, the characteristic functions for each component are multiplied. There are also several other aspects of the model that need to be optimized. One of them is the component parameters. So each component has a pattern covariance matrix, sigma K, representing a specific type of pleiotropic relationship. And these matrices have a set of parameters that are estimated using gradient descent. This is sensitive to initialization. So we have a procedure for the number of initializations and stopping criteria to try to reach an optimum. Additional other procedures that I will not go into detail with, but just wanted to mention, are that we iteratively fit the model and rotate the empirical characteristic function based on the current estimate of the effect size distribution so that we can eliminate sampling times that are correlated. Other details are how we chose the sampling times and partitioned SNPs into jackknife blocks for calculating the empirical characteristic function. And finally, regarding the computational limits of this method, we have found that it is fast for 2 traits, but it becomes much slower for 6 or more. So are there any questions so far?
There are not at the moment. Thank you.
Okay. So now I'll go on to some results. So we did a variety of simulations to test the performance of PDR. The first set of simulations were to compare PDR with 2 other methods, genomic SEM and DEGA, or SVD as we call it here, where SVD was implemented by performing eigendecomposition on the sample covariance matrix. We fit models consisting of 1 to 3 factor-like rank 1 components to a cluster of 6 T2D related traits. Then we simulated data from each of those models and fit PDR to those models to get estimated factor weights, which we call theta. Then for SEM and SVD, we used the true covariance matrices as inputs and applied those methods to get the factor weights. Then comparing these estimated factor weights with the true values, we calculated errors for each of the methods. So across all 3 models, we found that PDR was able to recover the true factor loadings and had smaller error than the other 2 methods. especially for the 2 and 3 component models. Genomic SEM and SVD were able to recover the factor weights for the 1 component model, since this component was able to capture most of the information to infer the factor weights. However, for the 2-factor model, SEM was able to partially recover the factor weights, whereas SVD was not able to recover them, and neither recovered the true factor weights for the 3-component model. Since we were applying the methods to data that had been simulated from PDR-based models, which potentially gave PDR an advantage, we also did some analyses to test whether this was the reason why PDR did better. So we also simulated data from models that were designed to favor SVD and genomic SEM. So here we can see that PDR was still able to recover the factor weights with low error. And for the results from the model based on SEM in panel A, genomic SEM was able to recover the factor weights, whereas SVD did not. And then for data generated from a model based on the SVD method, we can see that PDR and SVD recovered the factor weights, but SEM did not. We then applied PDR to clusters of real traits from the UK Biobank, including a 6-trait cluster comprising coronary-related traits and a 5-trait cluster consisting of asthma-related traits. For the CAD cluster, we identified BMI-related, hypertension-related, and cholesterol-related components based on the variance explained for each trait in each component. The BMI-related SNPs explained a large proportion of BMI variance, and they also affect each of the other traits. The hypertension-related SNPs mainly affect CAD and hypertension, and the cholesterol-related SNPs mainly affect triglycerides, HDL, and high cholesterol. These clusters seem to make sense, indicating potential distinct mechanisms underlying CAD that are related to these 3 clusters. Looking at the posterior scalars, which indicate membership in each component, we found that most SNPs belong to a single component, as shown in panel B, where most SNPs lie along the axes. Here we can see scatter plots of SNP effect sizes on some of the trait pairs. SNPs are colored by their membership in one of the 3 pleiotropic components or a trait-specific component. For visualization purposes, SNPs were assigned to a single component here. We see that BMI-related SNPs affect hypertension, but hypertension-related SNPs do not have much of an effect on BMI. However, the slopes for the CAD versus hypertension effect sizes are similar for both hypertension-related and BMI-related SNPs, indicating that the correlation between BMI and CAD might be explained by the effect of BMI-related SNPs on hypertension. This is consistent with a model where BMI is causal for hypertension, which is causal for CAD. On the other hand, we observe that hypertension-related SNPs affect high cholesterol, but cholesterol-related SNPs do not affect hypertension. And looking at the CAD versus high cholesterol slopes, the hypertension-related SNPs appear to have a larger relative effect on CAD than the cholesterol-related SNPs, suggesting that the hypertension-related SNPs have effects on CAD that are not mediated by cholesterol. So this is not consistent with a model in which hypertension would be causal for high cholesterol, and high cholesterol would be causal for CAD, in which high cholesterol would have mediated the effect of hypertension on CAD. We then applied PDR to the cluster of asthma-related traits, and we found BMI-related pulmonary and inflammatory clusters. Again, the BMI-related SNPs explain a large proportion of BMI variance while also affecting the other traits. The pulmonary SNPs affected mainly asthma and FEV1 FVC, and the inflammatory SNPs affected asthma, eczema, and eosinophil count. This supports previous findings that asthma has obesity-related and allergy-related subtypes, where the obesity-related subtype corresponds to adult-onset asthma, and the allergy-related subtype corresponds to childhood-onset asthma. Again, looking at the component scalars on the right, we can see that there was little overlap between the components. And here, scatter plots of SNP effects on pairs of traits in the asthma cluster are shown. So some things to note here are in panel E, we can see that the pulmonary SNPs that increase FEV1 FVC tend to decrease the risk of asthma, as indicated by the negative slope of the orange points. This indicates that high pulmonary function might be protective against asthma. We also note that there are non-transitive genetic correlations between BMI, asthma, and FEV1 FVC, where BMI is positively correlated with asthma and FEV1 FVC, but FEV1 FVC and asthma are negatively correlated. However, here we can see that BMI-related SNPs have positively correlated effects between BMI and the other two traits, as shown in panels C and D, whereas pulmonary SNPs drive a negative correlation between FEV1 FVC and asthma, as shown in panel E. So again, this shows how breaking SNPs into components that have different correlation patterns can give more insight into multi-trait genetic architecture than the overall genetic correlation. So before I go on, are there any questions?
There are not at the moment. Thank you.
Okay. Next, using the effect size distribution estimated by PDR, we can calculate posterior mean effect sizes to give better powered effect size estimates by pooling information across multiple related traits. We compared the predictions from a few different methods, including simply using the original summary statistics, as well as by applying PDR and MTAG, which performs linear meta-analysis using optimal weights. The MTAG estimates were obtained using the best linear unbiased predictor, or BLUP, which uses a multivariate normal prior, which is needed to calculate the posterior mean effect sizes, and has identical replication R² to MTAG. For asthma, T2D, and CAD, we were able to calculate predicted replication R², and for asthma and T2D, which had available out of sample data, we also calculated observed replication R². Additionally, we calculated variance explained, which gave us a measure of the replication R² for subsets of SNPs bin by their effect size. So overall, this analysis aimed to both validate our model as well as improve effect size estimates. So here we see that PDR had a higher replication R² than both the original summary statistics and MTAG, with a 94% improvement over the original summary statistics for asthma and a 70% improvement for T2D. PDR applied to T2D showed a much smaller improvement over MTAG. However, the expected and observed R² values were very close. Note that the validation data were normalized so that the expected and observed values were equal for the original summary statistics, which is why the observed R² is not plotted here for the sum stats. We did not have validation data for CAD, but based on the expected R², there was an improvement of 300% in replication R². This might be because the replication R² was smaller for the original CAD sum stats, and since the CAD data was not from the UK Biobank, pooling this data with the much better powered UK Biobank data within PDR likely allowed for a substantial improvement. Then here we observe the expected and observed variance explained for SNPs bin by effect size for asthma and T2D. We can see that for both traits, PDR is approximately unbiased across all SNP bins, whereas MTAG was unbiased on average, but tended to overestimate smaller effects and underestimate large effects. This is likely because the multivariate normal or infinitesimal prior used by BLUP does not enable a small percentage of SNPs to have large effects. So to summarize, PDR is able to infer shared components of heritability across multiple traits, as well as to characterize their pleiotropic mechanisms and identify the variants that belong to the different components. PDR also improves effect size estimates by pooling information across multiple correlated traits, and this boosts the correlation between the estimated and true effect sizes. And finally, Estimates are unbiased across SNPs with effects spanning the range of effect sizes that explain most complex trait heritability. So I would like to thank Luke O'Connor for all of his support and guidance throughout this project, as well as the other members of the O'Connor Lab. Thank you very much for your attention, and I look forward to any questions that you might have.